Okay, so we're going to have a look at DNA in a bit more detail. So this is one nucleotide. We've got our 5-carbon sugar, there's your 5th carbon, phosphate is attached to the 5th carbon, and the nitrogenous base is attached to carbon 1. This is another nucleotide. Remember, this is the only bit that changes, so this could be A, this could be C, it doesn't really matter. Now what we're going to do is we're going to join these together. Now remember, in the last video we talked about that being a glycosidic bond, we talked about that being an ester bond. The phosphate here is going to form another ester bond with this 3rd carbon. So that 3rd carbon would actually have OH on it. Remember, we're going to remove water, and so we end up with a bond between that 3rd carbon and the phosphate that is on carbon 5. Now, DNA polymerase can only add a new nucleotide onto this 3rd carbon. It can't add a nucleotide onto that 5th carbon. So it can only add a nucleotide onto the 3rd carbon end, which is known as the 3' end. It cannot add add another nucleotide onto the phosphate of this 5th carbon. So that is called the 5' end. So if you end up with a great long chain of DNA, if you want to add the next nucleotide on, you add it to this end. It won't add it to that end. So this is the 3' end. So you'd have the phosphate, carbon 5, the sugar, the nitrogenous base, and it just repeats. Now, as I said, that is called an ester bond. That is also an ester bond. So in the formation of the phosphate sugar backbone, and so we call it, very imaginatively, the phosphate sugar backbone. The bond involved in the formation of the phosphate sugar backbone is called a phosphodiester bond, because you've got phosphate, ester, ester bond. Phosphate, ester, ester bond. So what's making that sugar phosphate backbone is a phosphodiester bond, but that is made up of an ester bond between the 5th carbon and the phosphate, and an ester bond between the phosphate and the 3rd carbon. So 2 ester bonds with a phosphate in between. So in DNA you get your phosphate sugar backbone, you get your nitrogenous bases sticking out, and we have direction, 5' and the 3'. Now when we look at a strand of DNA, a double helix strand of DNA, very wonky double helical strand of DNA, if that end is the 5' and that end is the 3rd prime, the other strand runs anti-parallel. It runs in the opposite direction. So that is, if that's the 5' end on the other strand, that is the 3' end. And if that's the 3' end on this, then over here, that is the 5' end. Now remember, we can only add another nucleotide to the 3rd prime, there or there. So let's see how that happens. Now, our double strand of DNA, first thing that happens is that we unwind it and we break those hydrogen bonds, and that is the job of helicase. Helicase unwinds the helix and unzips the helix by breaking the hydrogen bonds. So then we end up with our 2 template strands. So the original double helix separates, so we now have 2 template strands. And we're going to make new DNA against that. Now, the DNA does not just all peel apart. It would all get into a very tangled mess because it's very, very long. So what happens is helicase unwinds and unzips as it goes along, adding in new bases and forming new DNA, 2 new strands, as it goes along. It doesn't just unwind the whole thing. So let's have a look. If that's the 5' end, that means that's the 3' end. If that's the 3' end, this is the 5' end. Now, let's just put in a base. Now, I am going to draw I'm going to draw the right thing. The base, I'm just going to do a coloured circle for the sugar. So that's the ribose. Now, where is our phosphate going to be? If that's the 3' end, that means our phosphate is sticking up here. So that's the 5' end. So if that's the 5' end, this is the 3' end of this particular nucleotide. So that means DNA polymerase can add in the next one. So you get the next nucleotide, and again there's the 3' there, so you can add in the next one. And your DNA polymerase will be doing that phosphodiester bond there, phosphodiester bond there, so doing those ester bonds between the 3rd carbon and the phosphate, 3rd carbon and the phosphate, to make that elongating strand. So DNA polymerase can just keep adding in the next base, and you can end up just going along this strand, adding in new DNA bases, nice and straightforward. So this strand is called the leading strand because it adds in the bases, pen to work, continuously. So it is continuously going along and adding the next base, the next nucleotide, then add the next nucleotide, then the next nucleotide, then the next nucleotide, doing that ester bond to form that phosphate sugar backbone. Okay, now the other strand, that's different. So let's imagine we add in our first base. That's our deoxyribose sugar. If this is the 5', then that means this is the 3' end. Now remember that phosphate sticks up out from the 5th carbon. So that means it's pointing out that way. Now polymerase cannot add to this end. It can't add to the 5' end. It can only add to the 3rd carbon. So we've got a bit of a problem. It cannot add anything to this nucleotide. So what polymerase does is it jumps forward. So this is the 5th carbon there. This one would be the 3rd carbon. So we have a 3' end that is kind of free, if you like. So what happens is polymerase adds in the next one, like that, and then it moves to the next one. Adding in the next one, so it's doing that phosphodiester bond, but it's going in that direction. So this one just continuously added them in as it was going. This one jumps forward and then goes backwards. And when it gets to the end here, it loops and jumps ahead again. So we'll just add in these ones. So again, we've got a 3' end here, so we can add in the next one. And so we're going to continue to go down that way. Okay. Now, when it gets to this end, it'll jump ahead again. Now the DNA polymerase has done each of these reactions as it's gone in this direction. Jumped up, done each of these reactions as it's gone in that direction. But you'll notice we have these sections. Those aren't knitted together there, bonded together. Those aren't bonded together. There is a third enzyme. It is called ligase. Ligase knits chunks of DNA. So DNA polymerase does every individual nucleotide as it goes along. Ligase fixes sugar phosphate backbone. So it joins pieces of DNA together. Now, if you want additional information, those fragments that are formed and the fragments are bonded together by ligase are called Okazaki fragments. But all you really need to know that on this strand, which is called the lagging strand, it is the DNA nucleotides are added in discontinuously. They're added in sections, kind of going the wrong way. So that's why this is called the lagging strand. It's always lagging slightly behind. So, as DNA unwinds and unzips, you've got your bases exposed. You've got nucleotides in the nucleus. They're added in continuously on the leading strand, and so it will just run up that way continuously. On the lagging strand, it jumps ahead and goes in that direction, then jumps ahead again and goes in that direction, and again in that direction. Then when you get to the end, it'll start to unwind and unzip more DNA. So this is the leading continuous strand. This is the lagging discontinuous strand. And these segments are called Okazaki fragments. And along with polymerase doing every one of these bases, these pieces are knitted together by ligase. That's it.